Hi guys, my name is Ashish Kumar Jha and I am Assistant Professor at Hislop College, Nagpur. In this class of cell biology, we will be studying reaction of glycolysis, oxidative decarboxylation of pyruvate and citric acid cycle. Of the oxidation process are carried out by enzymes of glycolysis, which are located in the cytosol. Glucose is broken down to pyruvate with the net formation of two molecules of ATP and NADH. Under anaerobic condition, the NADH is reoxidized by the conversion of pyruvate to lactate. Under aerobic condition, pyruvate is further metabolized by the citric acid cycle. The citric acid cycle is the central pathway in oxidative metabolism and completes the oxidation of glucose to 6 molecules of carbon dioxide and water. In the initial stage, the breakdown of glucose is common to virtually all cells. Glycolysis occurs in the absence of oxygen and could provide all the metabolic energy of anaerobic organism. In aerobic cells, however, the glycolysis is the only first stage in glucose degradation. The reaction of glycolysis results in the breakdown of glucose into pyruvate with the net gain of two molecules of ATP. The initial reaction in the pathway actually consumes energy using ATP to phosphorylate glucose to glucose 6 phosphate and then fructose 6 phosphate to 1,6 biphosphate. The enzyme that catalyze these two reactions are hexokinase and phosphofructokinase respectively. The reaction following the formation of fructose 1,6 biphosphate constitute the energy producing part of glycolytic pathway. Cleavage of fructose 1,6 biphosphate yield two molecules of 3 carbon glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. The phosphate group of this compound has a very high energy, so it is used to the next reaction of glycolysis to drive the synthesis of ATP from ADP. The product of this reaction, 3-phosphoglycerate, is then converted to phosphophenol pyruvate. The second high energy intermediate in glycolysis. In hydrolysis of high energy phosphate of phosphonol pyruvate, its conversion to pyruvate coupled to the synthesis of ATP. Each molecule of glyceraldehyde 3 phosphate is converted to pyruvate is thus coupled to the generation of two molecules of ATP. In total, 4 ATPs are synthesized from each starting molecules of glucose. Since 2 ATPs were required to prime the initial reaction, the net gain of glycolysis is 2 ATP molecules. In addition to producing ATP, glycolysis converts two molecules of coenzyme NAD plus to NADH. In this reaction, NAD plus acts as an oxidizing agent that accepts electrons from glyceraldehyde 3-phosphate. The NADH formed as a product must be recycled by serving as donor of electron for other oxidation reduction reaction within the cell. In aerobic condition, NADH serves as an additional source of energy by donating its electron to the electron transport chain, where they are ultimately used to reduce oxygen to water, coupled to the generation of additional ATP. In anaerobic condition, the NADH formed during glycolysis is reoxidized to NAD plus by conversion of pyruvate to lactate or ethanol. In eukaryotic cell, glycolysis takes place in the cytosol. Pyruvate is then transported into mitochondria, where its complete oxidation to carbon dioxide and water yields most of the ATP derived from glucose breakdown. The next step in the metabolism of pyruvate is its oxidative decarboxylation in the presence of coenzyme A, which serve as a carrier of acyl group in various metabolic reactions. One carbon of pyruvate is then released as carbon dioxide. The remaining two carbons are added to coenzyme A to form acetyl coenzyme A. In the process, one NAD plus is reduced to NADH. The acetyl coenzyme A formed by this reaction enters citric acid cycle or Krebs cycle, which is the central pathway in oxidative metabolism. The 2-carbon acetyl coenzyme A with oxaloacetate, it is a 4-carbon compound, yield citrate. This reaction is known as a condensation reaction. Through 8 further reactions, two carbon of citrates are completely oxidized to carbon dioxide and oxaloacetate is further regenerated. During the cycle, one high energy phosphate bond is formed in GTP, which is used directly to drive the synthesis of one ATP molecule. In addition, each turn of the cycle yields 3 molecules of NADH and 1 molecule of flavin adenine dinucleotide, that is FADH2, which is another carrier of electron in oxidation reduction reaction. The citric acid cycle completes the oxidation of glucose to 6 molecules of carbon dioxide and 4 molecules of ATP are obtained directly from each glucose molecules, 2 from glycolysis, 2 from citric acid cycle. In addition, 10 molecules of NADH, that is 2 from glycolysis, 2 from conversion of pyruvate to acetyl coenzyme A, and 6 from the citric acid cycle, and 2 molecules of FADH2 are formed. The remaining energy derived from the breakdown of glucose comes from reoxidation of NADH and FADH2, with their electrons being transferred through the electron transport chain to reduce O2 to H2O. Thus, we will summarize the TCA cycle, where we see that acetyl coenzyme A is fed into the cyclic pathway where substrate is oxidized and its energy is conserved. One thing here we find that other than succinate dehydrogenase, which is bound to the inner membrane, all the enzymes of TCA cycle resides in the soluble phase of the matrix. The first step, that is condensation of the 2 carbon acetyl group with 4 carbon oxaloacetate to form 6 carbon citrate molecules. During the cycle, the citrate molecule is decreased in chain length 1 carbon at a time, regenerating the 4 carbon oxaloacetate molecule, which can condense with the another acetyl coenzyme A. It is 2 carbon that are removed during TCA cycle are not that are completely oxidized to carbon dioxide. During TCA cycle, 4 reactions occur in which a pair of electrons are transferred from substrate to an electron accepting enzyme. 3 of reactions reduce NAD plus to NADH, and 1 reaction reduces FAD to FADH2. Thus, TCA cycle is critically important metabolic pathway. And next class we will be taking about electron transport chain. Thank you.